Welcome to Learning with Philemon. In the previous video, we saw that some synthetic polymers are made by condensation polymerization, in which two different types of monomers join to form a polymer, and water as a byproduct. In this video, we will see that condensation polymerization occurs naturally also. Proteins are made in the ribosomes of cells. Proteins are polymers made from monomers called amino acids. Below we see two amino acids, glycine and alanine. Note that both amino acids have two functional groups, an amine NH2 group and a carboxylic acid COOH group. As seen in the previous video, when an amine reacts with a carboxylic acid, the nitrogen atom forms a bond with a carbon of the COOH group, as seen by the green bond. The OH and H groups lost form water. When two amino acids bond together, they form a dipeptide. Note that the end carbon and nitrogen atoms can bond with other amino acids to form a polymer chain. When more than 100 amino acids join together in this way, we get a protein. Note that the body uses 20 different amino acids in different combinations, not just glycine and alanine. At GCSE level, the structure of a protein will often be simplified to this type of diagram. The different colored boxes represent the carbon chains contained within different monomers, different amino acids. Note that like nylon 6,6, the synthetic polymer, proteins contain an amide link. Now let's see how proteins can be broken down. Proteins can be broken down by a process called hydrolysis. In hydrolysis reactions, water is used to break down a larger molecule into two or smaller molecules. If we react the dipeptide formed from glycine and alanine with water under acidic conditions, the amide link breaks, reforming the constituent amino acids. Because the reaction is conducted under acidic conditions, the amine groups of both amino acids are protonated. This means that the NH2 group bonds with a proton, an H+ ion forming NH3+. For more on this, please watch the videos in the Acids and Bases playlist. This process occurs not only for dipeptides, but for the longer chain proteins. If a protein is broken down by hydrolysis, the amino acids it contained can be separated and identified by chromatography, a separating technique. Now let's take a look at another natural condensation polymer. Starch, a complex carbohydrate, is formed in plants from the excess glucose produced during photosynthesis. Starch is a polymer made from the monomer glucose. Glucose has the following structure that consists of a 6-membered ring of 5 carbon and 1 oxygen atom. Note that unlike in proteins, only one type of monomer is used to form starch. When two glucose molecules react, a bond forms between the oxygen of one hydroxyl OH group and a carbon from the other glucose molecule, as represented by the green bond. Like in previous condensation reactions, an H and an OH group are lost to form water. When two glucose molecules bond, they form a disaccharide. When more than 1,000 glucose molecules bond in this way, we form starch. At GCSE level, the structure of starch and other complex carbohydrates will often be simplified to this type of diagram. Note that the green box represents the glucose monomer. In this case, there is no amide linkage. An oxygen atom links two glucose molecules together. Using particular enzymes in water, complex carbohydrates can be hydrolyzed just like proteins to form their constituent sugars, such as glucose. This is very much like reversing the following reaction, going from a disaccharide to two glucose molecules. Thank you for listening. To consolidate your learning, please watch all the polymer videos and try to answer the questions in the description. If you haven't already, please subscribe for more content. Stay curious.